In Chapter 5, we turn to looking at more of the specific properties of a cell and the specific functions of a cell. Chapter 5 is all about membrane transport, so moving molecules across that plasma membrane. The plasma membrane is really important because it separates the cell from its surrounding environment. So it provides a boundary that partially protects the cell, but mostly controls what moves in and what moves out. Because a cell is a living organism, it's not isolated from its environment. If it's a single-celled organism, such as a bacterium, it has to be able to sense its surroundings, sense the other living organisms around it, bring in nutrients, get out waste, and also it needs to move in its surroundings. If a cell isn't part of a eukaryotic organism, that cell is part of a bigger functioning unit, and so it needs to be able to communicate with the rest of the cells around it. And cells will also produce proteins and other molecular products that can be used outside of that cell in the larger organism. So the fact that the plasma membrane controls what moves in and out is really important. All biological membranes have what we call selective permeability. This means it allows some things to cross easily and other things are blocked. This is a way for a cell to control what moves in and out easily. There are several mechanisms that allow the passage of molecules in and out, and that's what we're going to talk about in this chapter. Some molecules can move passively through the membrane. Others must move through specialized proteins that allow them in or out. And other products are moved out by the fusion of vesicles. And also products can be brought in through fusion of vesicles as well. So we're going to talk about each one of those methods for moving things in and out of the cell. Membranes are considered a mosaic, so they're made up of a lot of different components. If you look at the very first picture in your textbook, there's a really detailed image of a plasma membrane. And I put different pictures here just to show you some of the different ways that it's represented and some of the different types of things that are present in the membrane. If you look at the first image in your textbook, there are a lot of rope-like molecules on the outside. Remember, that's the extracellular matrix. And then on the inside as well, we have the cytoskeleton. So those are giving the structural support, whereas the plasma membrane is giving a boundary. If you look at the picture on the left, we see a variety of brightly colored objects inside that plasma membrane. Those are different proteins, and each of those has a really specific function. We also see carbohydrates and other molecules coming off of the membrane. Those are involved in communication with the outside environment, sensing the outside environment, and some of those are structural, actually anchoring those proteins to the cytoskeleton or the extracellular matrix. And then we also see our cholesterol molecule in there. So those are really important in the cell membrane. We heard about those in Chapter 3, and we'll come back to those as well. This picture on the right shows a aquaporin. So an aquaporin is simply a protein that allows water in. So I like to remember it like water pours in. Those red and gray molecules are the water molecules. The blue proteins are the alpha helix of the protein. So that's a secondary structure of that protein. And the properties of that protein allow water in easily to flow through that plasma membrane. So plasma membranes are a really good example of emergent properties. They are proteins and fats that function together in a really ordered manner to help regulate transport across that membrane. So as I said a few minutes ago, cells exchange chemicals with their environment. This can be anything from sugars, amino acids, other nutrients, to waste products, gases such as carbon dioxide and oxygen, and then also ions, sodium, chloride, calcium, things like that. So let's review the structure of the phospholipid, because the phospholipid is the main component of that plasma membrane. Phospholipids are important because they are what we call amphipathic. Amphi means both, and the word amphipathic means that the molecule is both positively charged and therefore polar and hydrophilic, and then the other end has no charge. It's hydrophobic and nonpolar. And so that positive end works really nicely to interact with water, whereas the other end, the tails, work really nicely to interact with each other. And what this produces is something called a lipid bilayer, or a phospholipid bilayer. If you look at that image down on the right, we see those hydrophilic phosphate heads basically forming a layer on the top and the bottom, and then the hydrophobic tails in the center forming a layer there. This huge hydrophobic center is really what limits the movement of most things across that membrane. A lot of what is being moved in and out of a cell is hydrophilic, it's polar, it's charged. And so by having this large hydrophobic region, it really blocks a lot of those things from moving passively. Here we have the image that I was just talking about, the first image in your book. And again, these membranes are called a mosaic. A lot of times this membrane model is called the fluid mosaic model. Mosaic because it's made up of a variety of different parts, and fluid because all of those parts can move sort of laterally within the plasma membrane. So those proteins are not locked down necessarily. Some of them are anchored to the extracellular matrix, but a lot of those other components, the cholesterol, the phospholipids themselves, some of the proteins can move in a fluid manner. They can flow past each other. As part of this fluid mosaic model, because they can move, they also have to be stabilized in a certain way. These molecules will slide past each other, sort of like people in a crowd, but they don't move up and down. So if you think about people walking by one another in a hallway, nobody is moving vertically, they're all moving laterally. And that's because they're held together by hydrophobic interaction. All of those tails are being repelled by those hydrophilic heads, and so all of those tails want to stay together. This allows the protein to the plasma membrane to also interact with proteins. All of those proteins that are embedded in that membrane also have a hydrophobic region that allows them to stay in that hydrophobic layer. Again, this keeps them from moving vertically out of it because all of those hydrophobic regions want to stay together. This is just one more image. I just wanted to give you a variety of different images showing this fluid mosaic model. We're going to talk about some of these proteins. So here we have labeled the integral membrane proteins, protein channels, cholesterol. We're going to talk about all of these components specifically. One of the really important characteristics of a membrane is that they can change in fluidity. So fluidity just means the ability to flow, and that is due to the presence of unsaturated or saturated hydrocarbons. So just like other fats, your phospholipids can be unsaturated. And remember that unsaturated fats have a kink in them that keeps them from packing tightly. So in chapter 3, we saw this example as olive oil versus butter, or something like bacon grease. Olive oil is fluid at warmer temperatures because the unsaturated hydrocarbons within their makeup keep them from packing tightly. But when you have saturated hydrocarbons, they pack very tightly in neat little rows. And this allows the cells actually to change their fluidity depending on the situation. So the cells can change the ratio of saturated versus unsaturated fat depending on the environmental temperature. So as the temperature cools for a cell, those hydrocarbons slow down and begin to pack. And it's when they slow down and begin to pack that you get a more solid fluid membrane. You're never going to get a completely solid one, but you can slow down the fluidity by changing the temperature. So temperature will increase or decrease the fluidity, but cholesterol molecules are also really important in changing the fluidity of a membrane. So some organisms have, actually all organisms, have a membrane that is specifically adapted to the temperature in the organism, the temperature in the environment. And the cells can control that fluidity based on moderate changes by changing the composition of their unsaturated and saturated fats, and also by the amount of cholesterol that's in the cell membrane. So this is why we say that cholesterol is actually really important. Oftentimes people think of cholesterol as bad, but it's actually needed as a steroid, and it's needed in the plasma membranes. So cholesterol reduces the movement of the phospholipid bilayer at moderate temperatures. And it does that because it blocks the it blocks the phospholipids from moving in between the cholesterol molecules. So it sort of slows down their movement. But it does double duty. At low temperatures, when it's really cold, cholesterol actually blocks some of that really tight packing. So although it blocks movement at warmer temperatures, it also blocks a lot of packing. So depending on the environment, it can do both duties. Membrane fluidity is specific to the cell type and the cell environment. There are a variety of evolutionary adaptations that maintain an appropriate membrane fluidity. For example, fish that live in extremely cold environments have a high proportion of unsaturated hydrocarbons. That is going to prevent them from packing tightly. Some archaea and bacteria can live at really high temperatures. These organisms have unusual lipids that can withstand really extreme temperatures by remaining less fluid. So they have specific adaptations, so basically their cell membranes don't melt. Other organisms have the ability to change this composition based on time of year. So for example, plants that can tolerate extreme cold, like winter wheat here in Illinois, can increase their unsaturated phospholipids in the autumn and winter. So when they do that, they decrease the ability of them to pack tightly, and therefore their cell membranes are not going to solidify. So I'd like you to think about this question. Hydrophobic molecules dissolve easily in the membrane and pass easily. Why would a hydrophobic molecule move easily through that membrane? Polar and ionic molecules, including water, don't pass easily or quickly. So think about those things for a second. See if you can answer those before we get into some of the specifics of moving these molecules. So let's talk about membrane proteins. So proteins are really important because they not only give some structure and support to the membrane, but they also determine the membrane's function. So there are two major types of membrane proteins. The first are peripheral proteins. So these are held in place by integral proteins, the cytoskeleton and the extracellular matrix. Peripheral proteins are the proteins that are located on the outside or inside of the membrane. They're not embedded in that phospholipid bilayer. They help give kind of a framework and structure to the cell. And then we have other proteins, integral proteins, that are held within the plasma membrane. So integral membranes penetrate into and even across that hydrophobic interior. So they have a hydrophobic region around the center that allows them to interact and stay put within that hydrophobic region of the phospholipid bilayer. Integral proteins are really important because they are the proteins that are going to allow for the passage of other molecules into and out. And when they act as sort of passage molecules, they have to be transmembrane proteins. This means they have to span across the entire phospholipid bilayer. They have to be open on either end to allow molecules to enter one side and exit the other side. So we are going to specifically focus on the proteins that regulate transport. These guys are really important because they allow molecules that normally do not interact with the phospholipid bilayer or cannot interact with the phospholipid bilayer to move across that membrane. So they effectively allow molecules to avoid coming in contact with that phospholipid bilayer. There are a couple of different types that we're going to talk about. There are channel proteins, and these are a hydrophilic tunnel that allows polar, charged hydrophilic molecules to move through. Aquaporins are a good example of this. They are specific for water. Channel proteins may or may not be specific for a single type of molecule. It really just depends on what their function is. Some are going to be specific, some are not specific. And then we have carrier proteins. So carrier proteins are a little bit different in that they are always specific for a certain type of molecule. And the reason that they are specific is that they allow a molecule to bind. When the molecule binds, it causes a shape change and allows that molecule to pass through. As molecules are moving through a plasma membrane, they can either move by passive transport or active transport. So passive transport is a type of transport that does not allow or does not require the use of any energy. In passive transport, your molecules are going to move by diffusion. And we'll talk about diffusion in just a minute, but basically it's movement from an area of high concentration to an area of low concentration. They pass through a channel or a carrier protein, or even straight through that lipid bilayer, simply based on their differences in concentration gradient. So diffusion can move straight through a plasma membrane. It can also be facilitated. That means that there's a protein that is helping this molecule through, whether that's a channel or a carrier protein. The second way that molecules are moved through a membrane is by active transport. In active transport, you are still using proteins, but those proteins require energy to move the molecule. These are often carrier proteins who bind specifically, produce a shape change, and then move the molecule across the membrane. Sometimes they are pumps. We'll talk about those. And sometimes they use co-transport, which is where the movement of one molecule drives the movement of another.